welcome to Leah and others who will be joining us later to our brand new developing research skills. We are super excited to be offering this. And we're doing this, our office, the Office of Service Learning and Undergraduate Research, as you see there, has developed this with the wonderful Merit Scholars. And Ms. Levi, I think you are with us still. There you are. So Haley Levi here is, along with Keegan Curry, have worked with me to develop topics for students who've already started research. Is kind of our target audience, but even if you haven't, that's okay. You're still welcome here, of course, because there's lots of great information. But Haley and Keegan have done a fantastic job suggesting and developing and finding presenters on what we call, again, developing research skills. So these are skills that are really important that you develop after you've kind of started a research experience. So things like, finding funding, how to get published, thinking about grad school, leadership ethics. So, a little bit higher level, we have an introductory level that you can actually take as well. We have a course that's developed that has that 0 credit. But this level, we're super excited to add this as kind of like the next step on the ladder for folks to kind of get these cool research skills. So my name is Gareth Jones, and I know I have a slide here. Do you want to go ahead and put the next slide there, Nilu? So introduce ourselves, and then I'm going to have Haley introduce our speaker. So here I am. I'm in my, just finished my 5th year here at UAB in the Office of Service Learning Undergraduate Research. I'm the Assistant Director. We've got my contact information there. If you have any questions about undergraduate research, I'm here to help in any way that I can, virtually right now, hopefully in person in the future with you, but we're super excited to be offering this. We'll go to the next slide now. All right, Neela, you want to say a couple words?
Yeah, hi, my name is Nilu Varghese. I'm a VISTA in the Office of Service Learning and Undergraduate Research. If anybody's interested in getting more involved with service learning or has any questions about getting involved with undergraduate research, I'll be happy to help you. And with me, there are also 2 other VISTAs who are joining me. So, Destiny, do you want to go and introduce yourself?
Yeah, hi, I'm Destiny. I also work alongside Nilu and the other Vista Kyla in the Office of Service Learning and Undergraduate Research.
Hi, I'm Kyla. I also work with Nilu and Destiny and Gareth. And if you guys have any questions, you can always reach out to me as well.
So just to give a quick idea of what we do in our office, if you're interested with service learning, we can help you get involved with Blazer Kitchen or the Clinton Global Initiative University, which is basically if you have a small project you want to get involved with and you need a little bit of funding, this is a great opportunity to apply to. We also help with other things like Peace Corps prep or course support for service learning courses and other things like that. On the undergraduate side, we have undergraduate research ambassadors, research workshops like this one, and we also help put together the UAB Expo, which is actually happening really soon. So if you're interested in joining the UAB Expo, it starts April 19th, and I believe you have to submit an abstract by April 7th. Would that be right?
April 9th.
April 9th. Yeah, and we would be excited to see you guys there. If you're interested in applying for a job, we also have AmeriCorps Vista positions that are open, and you can go to our website for more information about this. And of course, if you want to be kept updated about different service learning or undergraduate research opportunities, feel free to come to our website and register to be sent the newsletters for service learning and undergraduate research. And yeah, if you guys have any questions or want to stay updated, follow us, like us, tweet us on our social media, and we'd be happy to get back to you.
Thank you so much, Neela. Appreciate you running through those slides. And we won't do this every time, but since this is the first of the series, we just wanted to let people know what our office is and some of the great things that we can help you with. But now I'm going to hand over to Haley, who's going to introduce our speaker for today.
Hi, I'm Haley. First and foremost, Keegan apologized for not being able to be here. He had a prior appointment he had to get to. But I am happy to welcome in Will. Will got his PhD at Florida State University, and he's now a postdoc in the biology department and is part of the PERSIST program. But yeah, Will, you have anything else to say?
No. So I'll introduce myself a little bit more, but I just want to say thank you to Haley and to Keegan and to Gareth for inviting me to give this talk. And thank you all for coming to watch. And so I hope that, you get something out of this. I know, everybody who's going to be attending this or watching this is going to be somebody who's already kind of crossed the first hurdle, which is to get interested in science and interested in doing something where you're going to be doing an academic pursuit or a research pursuit. And so I'm hoping that this gives a little bit of insight into sort of the challenges and the excitement and a little bit of background into how to do experiments or how to think about doing experiments. Mainly because I think talking about this early on in your education and early on in your career is a good way to save yourself a lot of pain later, which is why I titled my talk, How to Save Your Sanity by Making Sure You Use Good Experimental Design. So the goals that I have for this talk today are mainly to kind of talk through the purpose and scope of what we term experimental design and sort of the things to think about when you're going to do an experiment. And then I've sat down and thought about, if I wrote out a list of 10 kind of best practices for doing good science when you're thinking about experiments, I challenged myself to kind of list out what those things are. And so I want to share with you what I came up with about the 10 things basically I wish I knew when I first got started in research, in hopes that I can save you some heartache and trouble. And then I'm hoping we can have a little bit of a discussion about maybe experiences that you've had with research, questions you have about research. And then if we have time, we'll do a little bit of practice in thinking through the experimental design process. Okay. But you might want to know, who am I to tell you what to do? What background do I come in with that makes me, an expert on this? And I'll tell you, I'm still, every day that I go in and I do experiments, I've been doing this for a long time, but I'm still learning how to do this, right? This is a lifelong pursuit to get good at asking good questions, designing good experiments, and taking a project all the way from the idea stage all the way through to the publication stage, right? This is something you get better at all the time, but it's not something that you ever feel like you've mastered because it's a big, complex task that we're setting out to do. But I'll tell you a little bit about my background. I have a PhD. My current job is here at UAB as a postdoctoral research scientist. In the biology department. I'm employed through the NIH IRACDA MERIT program, which is a postdoctoral program that basically funds people like me to come to UAB or to another IRACDA program to do part research and pursue our careers there, but then part doing teaching and outreach so that we learn more of the skills that are what you would want to do if you go on to be a professor. And so being a professor is a lot of different kinds of jobs. And so this is a postdoc program geared toward that. In case you don't know, because I didn't know when I first started out what a postdoc was, essentially when you're going to become a scientist, the progression that you do is you go get a bachelor's degree, and then maybe you do a master's degree, and then you do a PhD, or maybe you go straight to a PhD, which is thought of as a terminal degree. And then after that, you're no longer a student, but there's another level of sort of training and experience that you can pursue beyond getting your PhD that helps you become more ready to step into a professor role or to work for the government or work for industry. And so it's typically a research-focused program, which allows you to sort of gain independence as a researcher without all of the pressure of being a faculty member. And so that's a typical step after a PhD, but before you step into what is going to be your final job. And that can be anywhere from a year to many, many years, depending on kind of what your path is. And so that's the stage I'm at right now. The field that I work in is marine evolutionary ecology. And so you might wonder at UAB why a marine biologist is there. We actually do have quite a few marine biologists on the campus, on the academic side in the biology department. And the primary thing that I study is trying to understand the role that environmental variation, so you think about temperature change or salinity or any of these environmental variables, how that variation influences the evolution of complex life cycles. And I use macroalgae and marine invertebrates as the study system that I use to ask questions, including some of these critters that are over here in the pictures. But I started off my journey as a bachelor's student. I got a bachelor's from California State University, Northridge. I'm a native of Los Angeles. And then I stayed there and started my research career, earning a master's degree there. And then eventually moved to Florida State University, where I earned my PhD before coming here to UAB. Okay, just to get started, so now you know me now. And so now I want to get a little bit of information about you who are listening to me in person, so that we can kind of structure the discussion and I know a little bit about you so I can know how to help you best. And so I want to try and use this service called Poll Everywhere. And so if you're somewhere where you can log into a website, you can log into this web address that's at the top, pollev.com/ and then you have this Will Ryan 348. And you can log straight in and it'll show you this screen. Or if you're just using your phone, you can text this Will Ryan 348 to this number 22333, and it'll log you into this session so that you can respond to the questions on the screen. And so I'll give just a minute for all of you who are listening to go ahead and log in to this survey. If you don't, if you're not able to log in, you can also just tell me, since there's, a small handful of you here. But I thought it'd be a good thing to understand kind of what experience you think you have with research so far, so that we can get into the brass tacks if you're, an expert already. Okay, great. It looks like some of you are having good success here. Okay, cool. So it looks like from what's coming in so far that most of you don't consider yourself experts already, which is great, right? I hardly consider myself an expert, and I've been doing this, through multiple degrees, right? And so, but it's great. We have a range of people who have never done an experiment before, people who have helped out with research. So oftentimes as an undergraduate, if you volunteer in a lab or if you get a job in a lab, you'll get put onto a research project and you'll do a piece that's this big without learning kind of the broader scope of what you're doing. And then we have, a good handful of you have done sort of, a little bit more in-depth work. So good. So the other thing that I wanted to ask you with using the same device is what you're interested in. And so this might be, your degree program, but you can enter as many different things as you want to into this, and it'll generate a word cloud of what everybody's interested in, just to give us all an idea of kind of the universe that we're in terms of our discussions. Cool. Education. Cool. So we've got a spectrum here, right? So we've got a couple of big votes for biology and for neuroscience, right, all the way to medicine, education, and we have a film studies person here too, right? And so I hope what is going to happen in this is that the type of skills that we're talking about developing by thinking about experimentation are going to be applicable for all of these different areas, right? Even if you're thinking about how to better approach a survey that you're putting out to generate interest from your community about something that you're working on, right? You can think through the structure of a project in the same way that a neuroscientist would think through running an electrophysiology experiment, right? The principles of experimental design and project planning are actually the same no matter what subject you're approaching. And so I think everybody should be able to get something out of this discussion. And I hope at the end we can talk a little bit about kind of where you're at in your thinking about these things, right? And so I'm actually not entirely sure how this survey is going to come across if you're using a phone. I think you might actually have to go to the website on your phone in order to answer this one. And this one's a little bit more complicated because I want you to sort of put in order which of these topics that are related to doing experiments you find most scary or stressful. I know I have an order in my mind, so I'm curious about what order you're going to come up with. All right, so we'll just give just about another 30 seconds here to make sure everybody's been heard. Okay, and so I will confess that I think my order of anxieties on this list of topics is probably exactly similar to what you all are talking about, right? You know, the big challenge of doing research, you know, coming up with ideas and thinking through kind of exciting things, right? Sometimes that can be challenging, but it's really taking a project from that exciting kind of nascent stage all the way through, you know, figuring out what to test, coming up with the specific thing you're going to do, and then generating and organizing all that data, right? And then the really, really hard part is turning that into a product that you can communicate to other people. The great thing is that it sounds like a lot of you are already really comfortable talking to other people about what you're doing, which is great, right? That can be a huge hurdle. And that's some of the best feedback you're ever going to get about your research is going to be talking to other people about it. But the thing that's a long-lasting product that you're going to produce is that written document that you're going to then try and get published. And yeah, oh man, that can be really stressful. But what I hope that you take away from what we're going to talk about today is that that process, that end product that you're producing, making that easy to produce starts at the very, very beginning of what you're working on, right? From the planning stage, if you have good experimental design and you're planning out what you're going to do and you have a clear idea of why you're doing what you're doing, when you go to write it up, you have already done the intellectual work that will help you outline what you're doing and build your argument, right? And then at the end, you're just assembling the thing that you've already built. And so that's really the goal of doing good experimental design, is making it so that by the time you get to the end, it all falls together the way that you planned, right? So the intellectual work happens at the beginning and not that hard lift at the end. Cool. Okay, and so I hope that you are you know, limbered up thinking about this stuff, because as we go through this, I want you to feel free to go ahead and unmute yourself and chime in and answer questions and ask me questions at any point here. It's a lot more interesting if it's a conversation for everybody, right? Okay. So what is experimental design? I've said that phrase like 16 times already, right? And so experimental design is really just the process of planning the logistics and analysis of an experiment or of research activity that you're going to do. It doesn't necessarily have to be an experiment. But the goal of that process of planning is to ensure that the data that you're collecting through your research is done ethically, it's done efficiently, and most importantly, that it's done with some foresight to how you're going to use that data downstream, right? The worst thing in the world when you're doing research is to just, give up whole swaths of your life in the lab counting something or out doing surveys or talking to people and collecting data and being very meticulous about every step, only to get to the end of that process and realize that you asked the wrong question. You wrote the wrong thing down. You measured the wrong thing, right? And then all of that work goes out the window because you didn't think at the very beginning about what you were going to need to analyze at the very end, right? And so the whole goal of experimental design is to try to develop that foresight to think through the whole process so that when you get to the end, you have what you need because you can't go back, right? And so the whole goal, right, is to put in the work now to make sure that the hard work amounts to something later. Okay. And so when you go through these kinds of steps that we're going to talk about, the benefits of that process is that it's going to allow you to produce a detailed supply list, protocol, and timetable so that you have a very solid idea of where you're going, what supplies you need, how long it's going to take you, how much help you need to bring in, right? By building out that kind of project plan, you're going to make sure that that experiment runs smoothly, right? And very essentially, especially if you're working in something like neuroscience, right, that you have that plan laid out well enough that you can go and get the proper permissions, that you can do that work ethically, right? Good design is going to allow you to anticipate problems before they start so that you can plan alternative strategies for things that you know are probably going to go wrong, right? So not only can you avoid pitfalls, but you can build in plan B things because you know at some point something that you're going to do is risky, and so you have a plan B or you have extra supplies. For me in my research, this is like having an extra pair of rubber boots because I know I'm going to step on an oyster and cut a boot open and have to get extra boots, right? And so I know where the fall in the plan is going to happen, and so I can prepare for that. If you don't do good planning, you're going to get out there and then you're going to be walking around in the socks in your mud, walking around in the mud in your socks, and nobody wants that, right? So this design process is going to let you compile a detailed record of how you did things, and most importantly, why you did the thing you did, right? We're going to talk a little bit further on about the importance of good note taking, but you want to record how things were done, why things were done, because at some point you're going to have to deal with this data you've collected, and you're going to have to write up what happened, and it might be far in the future, right? A project you're working on today in a lab as an undergraduate, you might be writing up that project with your PI, with your mentors, like 5 years from now. It seems crazy, but that's how these things go, right? In 5 years from now, you're not going to remember the details of what you did. And so good planning at the beginning and the note taking throughout is going to allow you to keep track of all those details and know why you did what you did and what you did, right? And then finally, this design process is going to allow you to produce robust data that actually addresses the question that you're interested in. Again, the second worst thing that can happen, other than having totally useless data, is having data that you realize doesn't actually address the thing you thought you were testing, right? And so good design and a lot of forethought before you start doing the fun parts is how you avoid missing the point with the data that you collect, okay? All right, and so these are sort of the 10 things that I came up with to think about good design. And so this is a participation point. And so I expect at least one person to be brave and unmute themselves. And so the first thing that you want to do before you even start thinking about supply lists and whatever is to identify very specifically the question that you're trying to answer. right? And this seems obvious, but when you start to ask questions, and then you ask questions and talk about them critically, you realize it's very easy to ask a very vague question, and you might launch a whole, multi-million-dollar research effort off of a question that you realize later was too vague, okay? And so I have a question here that's, what's the effect of a change in the minimum wage, right? That's a valid research question. It's a valid thing to ask if you're like a sociological researcher, an economic researcher. What's the effect of a change in the minimum wage? And so what criticism can you offer me? If I came to you and said, you have money, you're a grant agency, I want to find out what the effect of a change in the minimum wage is, what would you tell me as a reviewer?
I'll chime in if no one else is. Maybe the type of effect, if positive or negative.
Right, and so I said effect, right? Effect could be positive, could be negative. What else?
I think it also depends on who exactly are you talking about, like in one area, because in different areas, in different countries, they have different minimum wages, they have different environmental factors that go on in different, areas and, that sort of thing. So when you just
Right.
say the change in minimum wage, it's like, where are you talking about and who are specifically are you talking about?
Right, I mean, that's a great point, right? The minimum wage, I might assume that everybody knows I'm talking about the minimum wage in Nigeria, right? Because that's my area of research and I've written this grant, and your expertise might be in Italy. And so you think, well, I don't know, what is the effect of the minimum wage in Italy, right? And so being specific about what you're asking and sort of, if you're a biologist, what taxa you're talking about. Are you talking about humans? Are you talking about mice? Are you talking about zebrafish? Right? And so that's a really good point. Specificity is important, right? We could modify this and get at one of Gareth's criticisms, right? And say, oh, we want to specify what the effect is on, right? An effect is vague, right? But we could ask, oh, well, how does the minimum wage influence the economy? And I'll say, in the United States, just to make it more specific. Now, how would you criticize this? What could be better?
I guess what part of the economy? Like it seems very, very nitpicky, but what part of the economy do you mean? Like, do you mean how it affects, how people buy food and things like that? Like how much they can afford? How are you talking about like taxes wise, that sort of thing? Like what part of the economy like do you actually mean?
That's a great point, right? If I asked you to define the economy right now, if I had each one of us write down a definition for the economy, we would probably all write down completely different things, right? Because the economy seems specific because it's the economy. You hear it on the news constantly. But as you pointed out, every one of those things that you mentioned, every one of those variables that you mentioned are part of the economy, right? And so it looks like a specific question, but it isn't. Right? How does the minimum wage influence the economy? I don't know. Are we talking about buying power, right? And so we can go a step further, right? We can try to narrow down that variable of the economy and say, no, specifically what we're talking about is gross domestic product, right? How much does it change? How much does the gross domestic product change after an increase in the federal minimum wage, right? Because we could have been talking about state minimum wages this whole time. And so, this particular question still falls under vague because it doesn't specify what country we're even talking about. But at least we're down to a variable that's a specific measure, right? It's a specific index that you could go look up, right? The GDP. And so that's the point here, right? Is that each one of these questions, if you just asked it in sort of a conversation, people would accept it as a good question, right? What is the effect of the change of the minimum wage? But what you're doing when you're talking about a question is you're often filling in missing information with your own ideas, right? And so you accept that question from somebody because you have an idea of what the economy is, you have an idea what the minimum wage is, and you kind of fill those things in. But when you're a researcher, you don't want to leave anything to the imagination because those are the places where that vagueness is going to cost you later on because you didn't define for yourself what you're talking about, right? Right. And so this is you want to get to the brass tacks of what your question is. And part of that is about identifying what variable you're talking about, right? And so the second sort of piece of advice, the first one is ask a good question and figure out what question it is you want to ask. And the second one is within that, specify those variables of interest, right? And so in the last question, the GDP was the response variable that we would measure. That's the thing that's going on the graph, right? And so you want to figure out what the variables are that you're measuring so that you know specifically what to look for and how to design your experiment. Okay, so now let's say we have a research question, which is, how does the size of a blue whale influence the amount of food that it eats, right? We're in a totally different realm of science, but we have this blue whale. And so somebody help me understand what the variables are that we're talking about here.
I guess like it would be what's the average size of a blue whale? What are kind of like, the different ranges, what counts as small, what counts as big, what kind of food does it eat? And does that kind of food change depending on its location? And then you can also think of what kind of other predators may be around, things of that nature that change, what kind of like food it may eat. Does it change depending on, if there's another predator that eats all that food, will it change that so then it doesn't eat as much? So I guess those are the kind of things that kind of first pop up.
Totally. I mean, you nailed it straight on, right? The two variables in the basic question are whale size and the amount of food, right? And you pointed out that both of those are vague. Like we've identified the variable, right? Whale size, and it's like, well, whale size, whale weight, whale length, whale measured in miles, measured in meters, measured in what, right? What's the essential metric that we're going to use to measure a whale that we're then going to compare among whales, right? We want to know what small whales are, what big whales are. We need a scale, right? And then the amount of food, right? You could go quantity, or as you pointed out, another question you could ask here is what kind of food, right? Does their diet change as you go from a small whale to a big whale? And so you have at least two different variables to consider, right? The amount of food, like the wet weight of the food, and the type of food, right? And then you've also brought up, so even within this very seemingly simple question, you have to think bigger about what are all the different things that could also influence this. And so if you were going to try and ask this specific question, right? Like, how does the length of whale scale with the wet tonnage of food that it eats, right? What would you do with that extra information about, who else is around and all of that kind of stuff? What would you want to do in your study to try and deal with that?
That is, I guess that's, I guess the main thing is, I guess, to see where exactly the most abundant amount of blue whales probably is, and then try and see, how much they eat without any predators or any additional, ones that are around, how do they behave by themselves?
Right. So what you're talking about is controlling for those other factors, right? Is you want to isolate so that you are trying to make those other factors not a problem, right? That those other factors are not influencing this relationship you're trying to measure, right? So this is the same thing, and we'll talk about controls in a minute, but what you're talking about is doing the same thing as saying, if you're doing a PCR or something, right? You put water in one of the wells that doesn't have DNA in it so that you can make sure there's no contamination, right? That's a positive or a negative control.
Right.
And so it's the same principle here, where if you're trying to isolate the effect of these two variables on each other, you have to think about all of the other things that could be interacting here, right? But the core thing that you have to do at this very early step in your research project is figure out which thing you care about, right? Which variable do you think there's a relationship between? Is it the amount of food? Is it the kind of food? Is it the food in the summer where all the whales are feeding? Like, what is the question, right? And that is a very complex thing, even though it seems like the simplest step. But good, you're like already a pro here. Cool. Okay, so we, like, let's say we've got, we know it's going to be whale length, and we think that it's the amount of food consumed, just like the total mass, regardless of what's in it. That is the important thing. We think there's some relationship here that we're really curious about. Now we want to frame a prediction, right? Before we start collecting data, before we go out and take our measuring tape and try and measure a blue whale, right? We want to figure out what our prediction is because, well, you tell me, why do you make a prediction in science? What's the purpose of a hypothesis?
I think it goes back to what you're saying before as being being specific about, what you're actually trying to measure. And that can also help you define, what am I actually measuring? What kind of, how do I want to measure it? Those kind of things. So being specific kind of sort of helps eliminate like, oh, maybe that can't happen, but this most likely definitely can. It keeps you focused.
Totally, right? Having that specificity helps you make choices. And one of the paralyzing things about doing science is that, especially, let's say you got $1 million to go do research and they said, great, here's your lab, go, right? You're going to walk in the door, you're going to see all the supplies, and you're going to think, I could do anything. I don't know what to do first, right? And that's where this whole process helps you whittle down all those choices and keeps you focused on the most essential thing, right? And so you're right that you are going to use your prediction and your hypothesis to keep you kind of honest about what it is you need to measure and things like that. But it also helps you understand whether you have a good grasp on kind of the information that you're rooted in, right? This is why you read papers when you're going to do an experiment, is that you need to have a background on how the world works and then which part of the world we don't understand that you're trying to identify, right? And so if you understand kind of the theory of what you're doing, you should be able to make a prediction about what you expect to happen so that when you collect data, if that confirms your prediction, cool, then maybe you're right? If you get totally the opposite pattern, you need to know that your framework of thinking about this is wrong. And so you want to make a specific prediction so that you know kind of where you think you're going and why you think you're doing this so that you know when you're wrong. Right, and so the next step here, we've got good question, we've got a good set of variables that we've identified. The next thing we want to do is we want to make an explicit prediction, and that often can be done in the form of drawing the graph you think you're going to get. And the benefits of this are twofold, right? One, it's that test of the theory that you think, do you understand the mechanism that you're trying to test? And the other one is exactly what you said. If I can't draw the graph here, then that means that either I'm not being specific enough in my question, I'm not being specific enough in my variables, or I don't know kind of where I'm going, right? And so what would you expect the line to look like? Or is this a bar graph? Is this a line? What would you expect the graph to look like in this whale example?
I was mostly expecting probably a line graph or kind of like a data plot almost, where it has the different dots and it's kind of like then it has a line going through it.
Mm-hmm. Yeah, so a scatter plot, right, where you have sort of X and Y axis, you have X and Y points, and then you have a best fit line going through it.
Right.
So what's the prediction that you would expect based on, how does whale size affect how much food it eats?
I would think, as whale size, as they get bigger, they, tend to consume more food.
Yeah, right, that makes sense. And so if you were writing out your prediction, right, you might just draw an upward line, right? I expect that the bigger the whale is, the bigger their mouth is, the more food that they need to eat. Boom, there's your prediction, right? So if you can do that, now you're ready to move on to planning your experiment, right? If you can't draw this graph for the thing that you're trying to understand, and you don't know what the graph should look like at the end of your experiment, you're not ready. You need to go back and think more, okay? And that's true whether you're doing a simple experiment like this, or whether you're doing some complex gene expression, whatever. You got to know what the figure is going to look like at the end, otherwise you're not ready, okay? Okay, and so now, great, we know exactly what we want to test. We want to know what the slope of that line is between the whale and the food, right? And now we're ready to get out there, start renting boats, get measuring tapes, whatever we need, right? And so what I want to encourage you to do, and I think this is hard because what you're often confronted with when you have a question that you want to test is that you're confronted with, the reality that things are hard and expensive, right? Is that maybe the ideal situation that you have would be to go out and measure every blue whale that you can find and, make it barf up the food it just ate and weigh it, right? That would be the ideal test. But you know that that's crazy and nobody's ever going to give you the money to go do that, right? And there's probably some like serious laws that you would break, okay? And so what you do often is you immediately start reducing down the vision to what's feasible. And then you ask questions from the position of what's feasible, right? As opposed to what the ideal situation was. But what I want to encourage you guys to do is to think about, if you had all the resources and there were no rules and you could do whatever you wanted to do, what would be the absolute ideal experiment, right? What would get you as close to that answer as possible, right? And you draw that all out and then you go, okay, how do I get from there to what I can actually do, okay? So maybe your ideal experiment is to bring a bunch of great white sharks, put them into tanks, and have a whole aquarium full of great white sharks that you're gonna, measure the oxygen in or something, right? And what is actually feasible is a tank full of guppies, okay? But I want you to think about starting by imagining the shark experiment, because that's gonna help you identify what the critical question and the critical variables are, right? If you start with the small experiment that's feasible, you might do a good experiment, but it's not going to necessarily test the question that you want, right? When you start big and then have to reduce it down, it's going to make you be creative about how to test exactly the variable that you want to test. Right, and so always start with this idea of the unlimited experiment and then whittle it down to a feasibility situation, right? Okay, I won't make you design the shark experiment yet.
Okay.
Okay, and so, right, and here then we get to sort of the brass tacks part. And if you're a person who likes kind of logic puzzles and things like that, this is a part that gets really exciting, right? Because what you want to do is design your experiment in a way that you're going to then be able to analyze you're going to be able to do the stats, right? And so has anybody had a stats class? Have you guys gotten there yet?
I took one, but it was a little rough. I will
Right.
not lie.
Nilu, have you taken a stats class?
Yeah, I took one a while ago and I agree. Rough. That's the best way to say it.
Yeah, I mean, and that's like a pretty universal, right? It's a really different way of thinking, and I think it turns a lot of people off. But you got to think about it like a puzzle, right? And so it's like, yeah, there's a bunch of memorization and there's some math and stuff, but what you're trying to do is solve this puzzle of, you have all of these things. You're going to go out and you're going to do some physical thing. You're going to, count cells, you're going to count whales, you're going to do whatever. And then you have to put the puzzle together and figure out how you can tell, what the effect of one thing on the other is. And so I think there's a way to think about statistics that's a little less stressful. And it's about this kind of puzzle solving thing. But what you want to do is you want to think about your experiment, what you're going to do and the data you're going to collect in terms of how you're going to analyze it at the end, right? Because you want to make sure that every action that you're doing is generating something that's usable. And the only way that you're going to be able to convince somebody else that the thing you found is true is to put it into a statistical model that actually demonstrates that there's, more than a random chance that that pattern that you found is real, right? And so you want to have in mind, if you're going to do an experiment, so like the example I have here is we want to know the effect of a particular antibiotic on, the growth of some cells, right? And so we have some petri dishes, they have some cells in there. We're going to look at the effect of the antibiotic in 2 different temperatures, right? And so this is what's called a factorial experiment. And so if you've had your stats class before, does anybody want to take a guess of what kind of statistical test you would run for this kind of a factorial experiment? This might be kind of an on-the-spot question.
Is this for a chi-square?
No, so this would be a t-test or an ANOVA, right? So it's an analysis of variance because you have the effect of one set of factors and then the effect of another set of factors, and you have sort of all possible combinations of those two factors, right? And so you're going to count up something about the cells, like how many cells there are, and then you're going to put that into an analysis of variance test where you're trying to figure out how much of the variation among those cell counts is explained by one variable, right, temperature, versus the other variable, the antibiotic, right? And so, yeah, so you could design experiments like this. If you were counting, a chi-squared test would be if you were going to do this kind of thing, and then you were counting, say, the number of cases of a thing, right? If you were out, you know, measuring something on the rocky shore and you were counting how many barnacles were this combination of conditions, that would be a chi-square test. It would look very similar to this. So good eye on that. But what you want to think about in this situation is how are you going to do this experiment in a way that's going to convince somebody that there's a difference between whatever variables you think there's a difference, right? And so in this situation, like what is the purpose of this saline solution? I listed this as a control, right? What's the purpose of a control in an experiment like this?
It gives you something to compare the results to, like it, so you don't think that, oh, hey, it changed, but it actually didn't.
Right, it's a baseline, right? And so the way we can define a control is it's a baseline of no effect, right? You need to know, if you're thinking about how cells grow, you need to know kind of, you know, how the cells grow normally so that you can know if they grow in some weird way when you put antibiotics on them, right? So like maybe there's bacteria in these cultures and you put the antibiotic on them and they grow really well. Okay, and so you need something to compare that to so that you can show that there's a difference. If you just put the antibiotics on the cells and the cells grew, you would just have some pattern of cell growth, right? You wouldn't know if there was an effect of the antibiotic or not without a control, right? And so you'll see in this example, you know, I've drawn it so that there are 6 little plates in every one of these combinations, right? And so my replication is 6, right? There are 6 replicates in each one of these conditions. Why, you know, that seems time consuming and that seems hard. Why wouldn't I just have one plate of cells in each one of these combinations?
I think it gives some variability because if something happens to one of them, like if anything goes wrong, then the whole entire thing is wrong. But since you have multiple replications and multiple, you know, times that you did it, then it limits, like, you know, so all the data isn't screwed up. You have at least 6 as backups.
Right, and so it's the purpose is twofold, right? One is like if you drop one of these petri dishes on the ground, your whole experiment isn't ruined, right? Because you can just get rid of that petri dish and you still have 5 left. But there's like a sort of a philosophical, you know, reason too, which is that not every single cell is completely identical, right? Even if they're totally genetically identical and identical in every way, you put them in a treatment, there's going to be some variation in how they behave, right? And what you're trying to show in an experiment like this is that the variation between treatments is greater than that natural variation among replicates within a treatment, right? And so you have to have, your experiment has to be replicated because the entire purpose of doing an experiment is to prove to yourself and to other people, right? To demonstrate the effect of something. And so you have to show, you have to have enough information to show that, you know, show concretely, kind of without a shadow of a doubt, right? And so the key that you want to keep in mind that's going to help you think about the stats part of this, even if stats are kind of scary, is that you want to design your experiment in a way that it's very clear for you to know if it worked, right? You're often trying to show something that you think is going to happen. And so you have to have some goalpost in mind that would say, you know, if it goes this direction, then I know that it worked, right? And so you can always ask yourself that question as you're setting this up, right? What's the criteria that I'm setting in this experiment that's going to tell me if it worked or it didn't? Okay, and that's what statistics are, right? Statistics are a way of setting up a criteria for whether it worked or not that takes your kind of personal bias out of it, right? And so we can think about this problem in a little bit of a different way. right? We can think about why we need replication by thinking about two ponds that have turtles in them, right? And in this situation, we want to know which pond has bigger turtles. We're going to measure the length, the snout to vent length in a turtle, which is the standard way to measure a turtle from its nose to its anus. And we're going to measure that in inches. And we want to know which pond has bigger turtles, okay? And I'm going to show you, we're going to pull out one turtle at a time from each pond and look at them. And I want you to tell me at what point you think that there is or is not a difference if we just go sort of one by one and pull out a turtle from each. Okay, so we go and we pull out one from A and one from B. We get a 7-inch turtle from one pond and a 6-inch turtle from another. Are these ponds different?
Yes.
Okay, how different?
Well, technically only by one, so it's very small. So
Right, so they're two. So would you say that like on average, these ponds are different?
no.
Okay, right. We don't really have enough information yet, right? Okay, so I pull out, the next turtle from each pond. Okay, now are these different?
Yes.
How are they different?
Now it's the difference between, oh, I can't count 3.
Well, so you have the red one are pond A and the blue ones are pond B, right?
Yes.
And so, but are they really different? If I pull out the next one, do you think the next ones are going to be, which pond has bigger turtles on average?
B? No. Oh, Jesus.
It's not clear.
Yeah, because
Okay, so I'll pull the next one. Okay, which one has bigger turtles?
B, right? Yes? No?
Yeah, a little bit. Would you stake, your money on that if I keep pulling turtles out of here, there's going to be a difference?
Nah.
That's the test. The test is, would you bet, $10 that there's going to be a difference if I pull 100 turtles out of here, right? And so at what point would you feel comfortable betting $100 that one pond is different from the other? Now, do you think there's a difference?
No, not by too much.
Well, so if we take the average of these, right, the average size of the turtles in the red pond is about 5 inches. The average size in the other pond is about 8 inches, right? And so there is a difference here. But the question is, how many turtles would you have to pull out of here before you would say, yes, these are totally different, right? And the answer is probably more than you think before you'd be comfortable that there's a difference, a real difference between these two ponds, right? And so this is what replication does you, because there's variation, right? There's variation among individual turtles, even within ponds that may be really different from each other. And so using multiple individuals randomly drawn from the same population, you're accounting for some of that natural variation. And then you can use statistical methods to distinguish kind of what the odds are that these are really the same distribution, right? So that's when, if you've ever heard of a p-test or anything like that, or a p-value, right? That's what you're doing, is you're estimating the odds that there's really no difference, right? And so when the p-value is very low, that means that it's very little chance that these things are really the same in real life. It's kind of a backwards confusing thing. But you can use a method like a power analysis, which allows you to look at the variance within the groups and then estimate how many replicates you would actually need in order to say that there's a difference, right? And oftentimes what you do when you're first starting out is do a pilot experiment, which is like a small scale experiment, just to kind of understand what the variance among things is so that you can do a power analysis and you can figure out how many turtles you need to draw out of these ponds before you can convince yourself that there is or is not a difference, right? Okay, I know we're getting up to the end here, so I'm just going to bang through these last ones. But what I want you to encourage you to do is to clearly map out the logistics all the way through. You got to think through the whole lifespan of a project. And when you first start out, if I said, I challenge you to go run an experiment on turtles or something, you would think about this part, right? You would think about running the experiment, how to run the experiment all the way through. But what I want you to start thinking about is that there's a whole pregame to the running of the experiment, and then there's a whole post life, right? So the steps of running an experiment start with planning, then start with mapping out your supplies, then's the actual experiment, and then you got to do stuff with the data, you got to analyze the data, you got to write the manuscript, and then you have to do this whole months-long process of shepherding that manuscript through the publication process. And so what I want you to take away is that success at the end of this, shepherding this through publication and getting reviews and publishing your work, begins with the choices that you're making all the way at the top end of this, right? The planning phase up here, and this might be years before you're to the end of this chain on a particular project, but you're not going to get to the end of that chain unless you do a good job at that beginning planning phase with all this stuff in mind, right? And then this whole middle bit, you have to be very committed to doing these steps right through this entire process and to being ethical and methodical through this whole process in order to have success at this end bit, right? And so as you're planning, you want to be realistic about what it's going to take to go through this whole process for the question that you want to answer, right? And part of doing that is to write everything down, right? Hopefully, if you're working in a lab right now, they've started training you in keeping a lab notebook. And everybody kind of gets that, like, yeah, you got to write down the data you're collecting. That makes sense. But you got to write down the data. You got to write down the methods that you're working with. You got to write down the name and origin of the supplies that you're using. What if something, what if a lot of cells that you ordered from a company are bad and you didn't write down that lot number, right? You got to keep track of all that stuff so that you can be the detective when something goes wrong and figure out what that problem was at the beginning. But more importantly, you need to write down what the reasoning behind your decisions was so that 5 years from now, you can go back to that notebook and go, oh, that's what I was thinking. That's why I did it this way, right? You're writing notes to your future self to help your future self out. And so you want to write down any observations you have, the name of who's present so that you know who to ask if there was a problem. If you're outside doing work, you want to write down the date, the time, the weather conditions, because what if that thing you saw that one time was because it was pouring rain and that's why you saw it, right? You can't start to piece together those patterns unless you write down things that you observe, and then you can piece it together later. When you have problems, write down the details of the problems, figure out why things failed, right? Write down when you have good ideas for future experiments, write that down, write everything down, because you're trying to write notes to your future self, right? Do your future self a favor, because you will not remember anything later. You're going to do all this stuff. You're not going to remember any of it, right? You got to leave yourself a breadcrumb trail to get back, okay? And as you're going through all these steps, always try to keep that first big question in mind because it's going to help you make decisions, right? You want to make conscious choices. When you have to change the plan, you want to make diligent, conscious choices that are respecting of that big question, because little choices along the way that change how you're doing and what you're doing are going to affect how you're able to write it up at the end, okay? Keep the notes on how and why you did things, especially when things change, and keep notes on things that fail, because that's going to teach you about what's going on in your system. And then early, you want to start working out an organization system so that you know where those notes are, right? Have a lab notebook, organize your files on your computer, put dates on things, like start organizing it, because you're going to need this information 5, 10, 15 years from now, and you're going to want to know where to find it. Okay, and last thing, stay curious, pay attention, right? All of you here are already doing the right thing that you need to do to launch your career, right? You're here, you're working in your various fields, like you're doing what you need to do, and you want to stay curious and pay attention, right? Even if somebody hands you an experiment to do, you want to think about everything that you're doing and pay attention. And so I'm going to tell you just a little story from my own work. I work on this sea anemone called Diademene lineata. It's invasive. It's from Japan. It's found all over the world now. It's got a very cool story. And I work on parts of its life cycle and all kinds of things. In the course of my regular work, I have to look at these things under the microscope because I'm looking at whether they're making eggs and sperm and stuff. And I noticed one day that there was this weird stuff on the surface of the sea anemone that looked like grains of sand. But when you zoomed in, it wasn't grains of sand at all. It turned out which I didn't know at the time. I just started writing down notes every time I saw it. Every time I saw it, I wrote down some notes. Yeah, there's that weird grain of sand stuff on the sea anemone. I just made another column on my data sheet, right? And it turned out several years later, I figured out that it was actually a protist, which is a single cell eukaryotic thing. It's a protist that lives commensally with this sea anemone. That now I have found that it lives all over the world with this sea anemone. And so this is an invasive sea anemone from Japan, but this protist lives on the Pacific coast, it lives on the Atlantic coast, and we don't know yet whether that came from Japan too, whether it was an invasive protist that lives with an invasive sea anemone. And so now there's a whole project that I'm working on to try and untangle this thing. And it turns out that this is probably an undescribed species. And so because I started scribbling notes in the margins of my real work, I've now found this entire story that I would have never found if I had just gone, huh, that's weird, and moved on, right? And so that's where being a good scientist is about observation and curiosity, and then being very methodical about how you choose to do things, right? So that you can get to the end of that pathway in any one project and have the words fall out of you because you already have done the intellectual work, right? Okay, I know I've gone over. I'm sorry for that, but I would be happy to talk to any of you about any questions you have.
A question I mainly had trouble with because I actually haven't started research in a lab yet because I'm trying to find a mentor and plan on reaching out, later this March.
Yeah.
But the biggest question I have and the biggest fear that I have is just trying to know like exactly what do I want to research because my mind is like, there's a lot of stuff that has been done and you don't want to, it's like my mind is like, do I want to do what's already been done? Is there anything I can find out more about? It's like that never ending thing of, okay, somebody's done this, how can I contribute?
Well, so you'll go from that feeling. And so that my advice to you is just to find anywhere that you can start working and just pay attention to yourself. And the key is you want to be working on something that you're passionate about. And so if you're enjoying the work, but it's not your jam, right, then you can try other things, right? You don't have to stay in the same lab. You don't always have to work on the same project. But as you go through and you're working with good mentors who are good scientists, you will go through a transition at some point. Probably, if you go on to graduate school, at some point you flip from everything's been done before and it seems like we know everything to, oh man, we don't know anything and there's so many questions, I'm never going to get to them in my whole life, right? And I don't know exactly why or when that happens, but at some point you realize, and it's just experience, like you just get in there and you start doing it and you kind of do what people are telling you to do until you start having your own ideas. And then when you start reading more papers and kind of figuring out what is it that we actually really know for certain, you find out it's so little and there's so much work to be done that I wouldn't worry about that feeling right now.
Okay, thank you.
Yeah, you bet. And then it's overwhelming for a whole other reason, right? It's just like, oh man, I'm never going to get through this work. Yeah. But what else? Does anybody else have any questions?
I have a quick question.
Sure.
So when do you know if you've gotten too specific with the question?
Well, I mean, do you have an example? Because I would say like when you realize, it's boring to every person you talk to, then you're too specific.
I mean, I was just wondering I was just asking because like, I was wondering, like, I've heard of different researchers, like different fields of research that are focusing on like tiny, minute things. And I was like, is that too specific or is that like?
So there's a difference between the kind of specific that we're talking about, which is once you have a question, you want to be specific about the variables that you're testing so that you know what you're looking for, right? The kind of specific you're talking about is that it's a very esoteric question, right? That the question itself only pertains to a small part of the world, right? And so that you want to be careful about that, right? This is why, if you're thinking about a question about a specific shark that lives in a specific habitat, And that's the scale of your question, right? You want to think about how that connects to the wider world. Or if you're thinking about this in, your C. elegans worm, why is that important? What does that teach you about the whole wide world, right? Because you could have a very specific, I'm working in this tiny worm that nobody cares about it on surface, but what I'm doing in that worm is curing cancer, right? Like that, the scale of how that translates, you always want to keep an idea about how the thing you're doing is connected to everything else. Partly because that's how you get funding, and partly so that you're making those connections for yourself. But within that, you want to be incredibly specific about the thing that you're testing, right? So that it'll help you make all these decisions about what to do. Does that make sense?
Mm-hmm.
Cool.
And one other thing, how long do researchers keep their lab notebooks?
Oh, forever. I had a job in between my master's and my PhD. I had a job at a biotech company in Boston. And it was like the lowest paid job you can get at a biotech company. And my entire job was to digitize old lab notebooks, which literally meant I sat in a windowless sealed room that had a fire suppression system that would suffocate you if a fire started. And I would go for as many hours as I could and just put the old lab notebooks from like the 40s and 50s in under cameras, and then take pictures of with the cameras, and it was digitizing the old lab notebooks.
Mm-hmm.
And so when you work for a company, like a biotech company, that's a legal thing. Like you have to be able to prove that those ideas happened at that lab for patent reasons and things. But I mean, you keep them forever. Like, we have Darwin's notebooks from the 1840s or whatever, like his handwritten notes on his boat. And so at an institution like UAB, they will probably keep your lab notebooks at the lab you're working in, right? And so that's like a whole library problem of theirs.
Thank you.
Yeah, of course. Okay. And I realized I didn't put my contact information on here, but I'm very happy to talk to anybody about opportunities at UAB or interests, or if you have research questions or life questions or whatever. I'm more than happy to, as are any of the Merit Postdoc Scholars, which is the program that I'm in. Feel free to email me and I can put you into contact with anybody that you might want to talk to. So.
Well, I want to thank you, Will, and I don't mind you going over because that was fascinating.
Good.
I really enjoyed that, and I love that how you stayed curious and saw that and about the protists. That's a new thing I learned about you, and that's really cool. That I know someone who discovered that protists. You never know. I mean, that's the beautiful thing about it. So, Haley, any last words from you before we sign off?
I just wanted to say thank you so much, Will, for doing this. Really appreciate it. Great presentation. I'm so sorry. My brain is dead right now.
Absolutely. I'm very happy to do it. So I wish you success with the series.
Oh, and for the person who was asking about lab notebooks, here's like all of my labs, like previous grad student notebooks.
That's what I'm saying.
They are all here. I've gone through them and read a couple before to find experiments I needed. So
right, that is a forever library. Yeah.
Lab rats or pack rats,
Yeah.
All right, and Leah, thanks for joining us. Thanks to the Vistas also for helping coordinate things. And again, we will be putting this up on our YouTube. And thank you all for your time. Really appreciate it. And thank you again to Haley and Will. Appreciate it. And we'll talk to you all later.